Okay, this is where we left off Monday. We were learning how exactly the DNA of the B cell receptor and T cell receptor gets edited, and specifically we were looking at light chain recombination in the B cell receptor. So that puts us back here at this lovely process, where the first step of this process is recognition between the RAG1 and RAG2 recombinase enzyme and these RSS signal sequences, the ones at the 3' end of the V region and the one at the 5' end of the J region in particular, since that's what's being shown here. Once these two things bind, it creates the loop structure, and that loop is defined and twisted kind of through the enzyme based on the spacing of those 12 and 23 base pair spacers that are found in those recognition signal sequences, the RSS sequences. So it creates this turn in the DNA, and it brings the V region and the J region physically really close together so that this next part, the chemistry part, can actually occur. The enzyme then cuts just one strand of the two, and chemistry takes over. The OH group of that one strand that got nicked attacks the phosphate group on the adjacent strand and creates a little hairpin turn. And then very similarly, over on the other piece of the signal end, where the DNA does not represent anything that we want to keep at this point, the same basic reaction is going to take place where that OH group on the opposite strand will attack the other phosphate group and seal those ends together and create what's called an episome or an excision loop that is going to be trimmed out of the DNA and then lost. And that's what's happening over on the other side in this part of the figure, step 4. So at this stage, we're actually done with the excision loop. This thing can float off and get degraded and recycled into nucleotides to be used to make some more DNA. But we do still have some work to do back over here on this hairpin turn piece that was created. So step 5 involves reopening that hairpin turn. And the enzyme that does that is the enzyme called Artemis. And there's 3 different positions that this enzyme can actually trim or cleave this particular piece of the hairpin turn. And that's at the top edge, right in the middle, or at the bottom. And so you've got those 3 arrows, 1, 2, and 3, pointing at 3 different kind of spots along that hairpin loop where the enzyme Artemis can actually create another nick in the DNA. Typically, not always, but typically, the most common location for Artemis to actually do this cleavage is at the 3' site, so the bottom edge. Kind of on the underside. So if it does that, you get this outcome where you've got one piece of DNA that's a little bit longer than the other piece of DNA. For that matter, you'd get exactly the same thing that would happen if you used the upper sequence and you cut at the top. It's just a slight difference in positioning and what exactly happens from here. So then you have addition of what are called the P or palindromic nucleotides. These pieces in this overhang in this 3' region are actually going to get filled in this space, this empty spot, by another enzyme. And so addition of these P nucleotides is one of the things that's not coded for in the DNA, but contributes to the receptor diversity. How many of those get added depends on exactly where Artemis cuts and exactly how many of these P nucleotides actually get added in. And then for the light chain, the last step is ligation of the V and J segments. So then we have to actually glue those back together after you create those palindrome sequences. So these are known as palindrome sequences because your initial sequence basically hairpin cleavage by Artemis occurs, and you end up with this sequence, TCGA on one side, because of where this enzyme cuts. So TC and then GA, as we loop back around, ends up with TCGA. The opposite end ends up with ATAT. So these get filled in with TCGA, and if you were looking at the opposite strand, you would also get TCGA, which is why they're known as palindrome sequences. One goes forward, one goes reverse. They're exact opposites of each other, but they match each other. And this is just a kind of a strange phenomenon that has to happen because of how DNA works. So if you sit down and logically think about it, it does become kind of apparent, but it takes a while and some understanding of the structure of DNA, that not just the double-stranded part, but the fact that it's all built on complementary base pairing, and you have to sit down and kind of maybe write it out for yourself exactly how and why that works out every single time to create these lovely little palindromic sequences. But addition of those extra nucleotides makes those regions a little bit variable in length and a little bit variable in sequence. So it wasn't, like I said, coded for in the DNA, but it's one way that we get increased receptor diversity. And for the light chain, for now, that's basically all the editing that's going to occur.
So would the same thing happen where it gets opened at that one position? Like
The same basic thing, you still get addition of palindromic sequences if you open at the position 1, the 5' overhang. Yes. If Artemis actually cleaves at position 2, you don't get those blunt end overhangs, and so you don't get addition of the palindromic sequences. So a lot of this really depends on where the Artemis enzyme actually creates that cut in the DNA. Which part? All of it?
No, the last part you just said.
A lot of the what happens here is dependent on where Artemis cuts the DNA.
It's a random where it decides to cut it.
It tends to favor cutting at the 3' site.
Okay.
On the underside. But it will also cut at the 1 site or the 2 site. At the top or the middle. Okay, so the last 3 steps of this process only occur in heavy chain DNA. What we just looked at was light chain recombination and the VJ recombination. The same first 7 steps occur for DJ recombination and then again for VDJ recombination. So these last steps only occur because, in heavy immunoglobulin heavy chains that will have that D region, the diversity region, present. Step 8 is another cleavage. So in this case, it's an exonuclease enzyme that cuts the DNA back open yet again. Why do we keep cutting open these little loops that we formed? This does not seem to make a lot of sense. Like, you've made the loop, great, you should be done, call it good, move on, right? Except this is really crucial for giving us that receptor diversity that we need to be able to address the number of pathogens that are out there. So when you reopen this loop in this case, you don't get palindromic nucleotides added, you get random nucleotides added. The enzyme that does that is called TDT. And these are not coded for in any template. It is literally random DNA garbage that gets inserted. I shouldn't call it garbage. I just mean that there's no basis for it coded for in the DNA. There's no template for it. There's nothing directing this. It's just whatever nucleotides happen to be present get used. It's truly a random process.
Aids to the adaptive immune system.
And this is what?
Aids the adaptive immune system.
It does aid the adaptive immune system, yes. This ability to add random nucleotides into an existing DNA sequence, it seems like a strange thing for us to actually have, and that TDT enzyme interests me a lot because of its ability to add non-templated nucleotides. Like, why don't we see that in other cellular processes, or do we see that in other cellular processes is something that I wonder personally, and I'm betting you all probably don't care.
I think it's interesting.
It may be interesting, but it's something that you can study in graduate school if it interests you enough, and we can pronounce it good enough to know that it exists for now and basically what it does. So there's an interesting problem with this addition of random numbers of nucleotides. Does this happen in frame? That is, do they fit as a codon? Like, is this in a group of 3 nucleotides or not? Because if you add 4 nucleotides or 5 nucleotides or 1 or 2 nucleotides that are randomly added, then that could actually shift your reading frame and make a non-functional protein. The last step in this process, once you've added the random nucleotides, is to seal the DNA back together. Again, there's a DNA ligase complex that does this. And that's it. We are done. It does not get reopened again at this stage. Yes.
It says that steps 8 through 10 only occur in chains.
In heavy chain, correct.
Why not the light chains?
For whatever reason, light chain recombination doesn't get the same treatment, and it doesn't get the addition of the random nucleotides. I suspect that it has more to do with that D diversity region and the joints between the VD and the DJ regions. That D diversity region tends to form an additional sort of structure or loop in this immunoglobulin heavy chain that we don't see quite the same exact structure in the light chain. So they're a little bit different in terms of shape because of this. But not enough for us to like sit here and try to map out. So that means that there are 5 mechanisms that generate antibody diversity in naive B cells. Naive, this is a special term in immunology. What this means is that the cell has never encountered antigen. So right now, hopefully all of your immune cells are naive, sitting there quiet, just waiting for a pathogen to come by and have not encountered any recently. They have not yet become activated B cells or T cells.
Is naive like never ever, or is it?
Naive is never ever.
So once they encounter one, they can't go back.
Once they encounter antigen, they can no longer revert to being naive. All right, so your 5 mechanisms for antibody diversity. Multiple gene segments. That's where we started off talking about this, and we ended up with a number of different, like there were like 45 heavy chain or segments for the V region and X number for the J region and X number for the D region if present and the heavy chain, etc. That gave us up to 1.2 million combinations. So the second step of this, or the second mechanism for antibody diversity, is heavy chain and light chain combinatorial diversity, because not only did the heavy chains and light chains have all of these little gene segments, but then you could put a heavy chain with either a kappa or a lambda light chain, and any of those possible combinations of the kappa or lambda light chains. Addition of the palindromic nucleotides during light chain or heavy chain recombination, the P nucleotide addition, after Artemis cleaves and opens the hairpin back up. Exonuclease trimming. This is usually going to occur at the junctions between VD or DJ, and this is part of steps 8 through 10. So if I backtrack a slide, I don't think it'll actually let me because I'm actually recording today. No, it will not let me backtrack a slide. But if you think back for a moment to the previous slide, when the exonuclease reopened that loop that had reformed, one of the things that happens is the exonuclease enzyme can actually trim off additional nucleotides. It can actually cut off some of those palindromic P nucleotides that we had previously added, and then add in the non-templated random nucleotides, which is done by TDT. And that is actually mechanism number 5. Gene segments, combinations, P nucleotides for the palindromic sequences, random exonuclease trimming during heavy chain recombination, and non-templated N nucleotide addition during heavy chain recombination. So those last two are exclusive to heavy chain only. All of this requires alteration of the structure of the chromatin in the cell. So think back to your cell biology knowledge. Chromatin is this structure of a combination of DNA and proteins, like histone proteins. And in cell biology, I know one of the things that I talk about are acetylation and methylation of those histone tails, and how this actually regulates how densely compacted the chromatin actually is. That becomes really important in this story of how we actually activate or silence these B cell receptor and the recombination events that actually take place. So RAG binding, the recombinase enzyme binding, is affected by epigenetic modifications on those histones, and that includes one specific epigenetic modification on histone 3, lysine number 4, and that's the addition of 3 methyl groups, so trimethylation. When you add 3 methyl groups onto that particular histone protein on that particular lysine amino acid in that protein, or in that that histone protein tail, this is a modification that is associated strongly with the activation of transcription. But in this case, we're not looking for transcription so much as we are looking for the ability for RAG1 and RAG2 to bind. Instead of transcribing, we're going to edit. But it's the same basic process that the cell uses for transcription of a gene when we want to do actual gene expression and transcription and translation and all of that stuff. So the addition of histone acetyl groups or methyl groups affects the accessibility of the enzyme and its ability to actually reach the structure, into the structure. So you'll notice how in this image you've got these densely packed histones wrapped, the DNA wrapped around those histone cores, and it creates these fairly tight little fibers. For the RNA polymerase or for the RAG1 and RAG2 enzyme to actually be able to fit in there, you have to loosen this up a little bit. It's like trying to work out a knot in a thread, or if you've ever worn a necklace that's just a simple chain and it got a knot in it, you have to pick that apart very carefully, and it takes a while. So you have to pick apart the structure of the chromatin a little bit and loosen this up so that you can actually get this RNA polymerase, or in this case, the RAG1 and 2 enzyme, to come in and actually start this recombination process. Those VD and J gene segments that we looked at, those pieces along the DNA sequence, they were separated by sometimes hundreds of kilobases. That's hundreds of thousands of DNA nucleotides. That's a fair distance. So because these things are spread so far apart, this also has to involve higher order chromatin structure that also plays a role. So one of the earliest events in this process involves histone acetylation. And this is another one that is generally associated with transcriptional activity, is addition of acetyl groups onto those histone tails. Hopefully that sounds vaguely familiar from cell bio. What this does in this case is it triggers that VDJ gene cluster to detach from the edges of the nucleus and migrate into the middle of the nucleus. And that goes back to the very first image of this chapter where we had that little red dot that started off on the periphery of the green circular thing, and then it migrated toward the center. That's what was happening in that particular image. Probably the most crucial histone modification is that trimethylation of histone 3 lysine number 4, which I've already mentioned. And it's this trimethylation that loosens the DNA and those histones enough for the RAG1-2 complex to recognize those RSS recognition signal sequences, starting in the DJ gene cluster. That's the one that's going to have the highest levels of trimethylation to start with, and that's why recombination starts at DJ instead of VD.
You said it detaches from the nucleus?
It detaches from the edges of the nucleus, so the nuclear periphery, and migrates in toward the center. So if we look at this image and we kind of think about it a little bit, that means that up where they're showing the RNA polymerase 2, if we just assume that that is actually going to be our RAG recombinase, it's managed to find one of those recognition signal sequences. Now it's got to find its partner recognition signal sequence on the other gene cluster, which is hundreds of thousands of bases away. So it's going to have to drag a whole other piece of chromatin that's very distant up to the same physical location and unwind a little bit of it too. And all of that has to happen so that the DNA can properly loop through that enzyme, and using those 12 and 23 base pair spacers get lined up properly for that initial nick to take place. This process begins on one allele on one chromosome. So at first, you only do this on one of the two copies of your chromosomes, either the chromosome you inherited from your mother or the chromosome you inherited from your father. If this recombination attempt is successful, cool, it stops there. But if it's not successful, if for some reason this process fails, let's say because you didn't get good recombination or you added an incorrect number of random nucleotides and your protein just doesn't translate and make a functional B cell receptor, then you can try again on the other chromosome. So you've got a backup copy that your cells can also use. If neither of these recombinations, if neither chromosome can produce a functional antibody, the cell dies. This is a critical process. At this point, what you have on the surface of your cell, I'm going to sketch a cell membrane there, is you've got your heavy chain recombination. Heavy chain recombination always comes first. And it is initially done after you get heavy chain recombination. It's going to actually use a precursor, light chain. If this thing recombined properly and if it's successful, what will happen is you'll get signal transduction down into the cell and you'll get activation and pro-survival signals. So if this thing isn't successful, you won't end up with this complex at the surface, and your cell can't send those pro-survival signals. which means eventually the cell will die basically because of something, it's called death by neglect. The cell doesn't receive any happy pro-survival signals, and eventually it just goes through apoptosis as a result.
I assume if like a cell had a mutation and it could no longer produce that, it would also die.
Yes, if a cell has a mutation and it cannot go through this process, or if RAG1 and RAG2 are missing and it cannot go through this process, you do not make functional B cells. In fact, RAG1 and RAG2 knockout mice that are genetically engineered to be missing these particular genes do not make B cells at all. They can't, because they can't do this process of recombination. So their cells remain naked and all of them die during development. It's a severe immunodeficiency. These mice are susceptible to almost all infections and they die really, really easily. You have to keep them in a super special sterile environment. This is actually a theme that we're going to see again and again. This idea that we get signaling through these receptors that is pro-survival when it's actually present. And that this actually allows the cell to survive. You get the same basic thing that occurs with T cells. Once you get all of those pro-survival signals, this triggers another event to take place. Lots and lots, multiple rounds of mitosis. So cell replication. Then you go back and you do light chain recombination. If your light chain is successful, it will actually replace your surrogate light chain, the placeholder light chain, or the precursor light chain that had existed before, to make a complete mature B cell receptor. In mice, it's actually going to try the kappa light chain first, and then the lambda light chain. Those are the Greek letters kappa and lambda. Ooh, that's a lot. So we know what happens if this B cell receptor doesn't get formed properly, the cell dies. What happens if this B cell receptor is autoreactive? What if it actually binds to tissues that are, say, like heart tissues? That would mean your B cells would recognize your heart tissue as being foreign and would start making antibodies against your heart. That would be bad. So if that happens, the B cell will actually try recombination again using the other allele on the other chromosome.
So all of this takes place before the stuff that we just did, like the steps 1 through 10.
No, this steps 1 through 10 take place first. That makes your heavy chain, the red thing that I drew up there. Then, if that is successful, you get signaling through the pre-B cell receptor, and that triggers mitosis and pro-survival signals. If after the rounds of mitosis, then you go back and you try to make a functional light chain receptor. If at any point you can't do these processes, the cell will either die or it will try to recombine using the second chromosome.
What about autoimmune disease?
Autoimmune disease is what happens when your immune system gets it wrong and you actually accidentally let cells out that are autoreactive.
So you go through 1 through 10 first for a heavy chain, and then if that's successful, you do 1 through 7.
Yes.
Okay.
So you start off with steps 1 through 10 for heavy chain. And if that is successful, you go through mitosis, and then you do steps 1 through 7 again for light chain recombination.
Also for the H3K4, you said that was histone and lysine.
Histone 3, K is the single letter abbreviation for lysine.
Histone
Histone and
and histidine the same thing?
histidine are not the same thing. A histone is a full protein that the DNA wraps around. It's one of these grayish colored cylinder looking things. And then this orange and the blue colored stuff, that's actually DNA. So these things are like drums that you actually wind DNA around to help condense it. Histidine is an amino acid.
Okay, I wasn't sure just for
The names are awfully similar, yeah.
well, they have the same one letter. So I wasn't sure.
Okay, yeah, and they do have the same single letter abbreviation. Yes.
Why in the picture does it have some of the DNA is orange and some is blue?
It's showing 2 different chromosomes.
Oh. Okay.
Or 2 different regions of a chromosome, but in this case, I think it's just 2 different chromosomes. All right. So if you take a chromosome and you've got all of the how do I want to explain this? All the DNA is wrapped up on those histone proteins. And if you think about the structure that was just shown in that previous slide for a second, again, if I could go back a slide, I would, but since I'm recording, it won't actually let me go back. If you turned that sideways, it wouldn't necessarily look like a completely round cylinder. It would actually have this sort of flower shape to it. Crummy looking flowers, but flower shape. I don't know if y'all have watched Velma at all, but I can't help but think of toward the end of it where they actually where Fred tries to draw the family symbol and it's the flowers, and that's where the flowers on the Mystery Machine come from, apparently. And spoilers. It's a horrible show. Don't watch it. It's bad. I watched all of it because it's kind of like, it's so bad it's addictive. Better off just not starting. All right, so these crummy flower-shaped looking things, these are called rosettes. And they actually are still DNA wrapped around those histone proteins. You're just sort of looking at the fiber end first. So like if you were looking down the length of it from above. What you have to do is you have to loop out these V regions, and then you'll notice that these D regions are way over here. We've got to drag those physically back over and basically be able to kind of superimpose them on top of those V regions. So you've got to get your V regions and your D regions or your J regions to lay physically close to each other and kind of be separated by only a little bit of distance so that your RAG recombinase enzymes can actually get that twisty loop thing to actually form. What you're doing is you're pulling 2 separate pieces of DNA together in a very small space and getting that little tiny twisted loop to form. And that's actually what these fluorescence images are actually showing. So what you've got is this blue colored background stain. We don't really much care about that. But in the middle here, what we've got are these reddish spots. And these are like the V regions and the D regions. These are active regions where the chromatin should rearrange. This is in an earlier stage and this is in a slightly later stage. And what you see is that all of these little red spots condense into 2 very distinct areas instead of being spread out. And what that tells us is that those particular regions of chromatin have moved so that they line up on top of each other. These regions are also a little bit bigger and a little bit more intense in terms of their fluorescence color, which also helps to confirm that. So those rosettes, you've literally, or you can think of them as having to stack the flowers on top of each other and get your V regions and your J regions to line up exactly so that you can actually go through this process. Okay, so back to our structure of our B cell receptor. Let's take a minute and label this because we actually should have an idea of what this thing is. Y'all have drawn these at this point and told me all about them. And yes, I'm going to work on grading those assignments, so I should hopefully have them done this weekend sometime. All right, so out here you've got the variable regions. Variable, that's the constant region, and these are the constant regions. Constant, variable, and constant. Okay, so we've got our variable and constant regions. All of this is determined by your VDJ and C recombination, or VDJ recombination at the variable tips. So VDJ, D for the heavy chain, so let me put that on this side, VDJ recombination. And on the light chain, you just have VJ on the recombination. Those constant regions on the heavy chain can be of one of 5 different types. Mu for IgM, delta for IgD, gamma for IgG, epsilon for IgE, alpha for IgA. Yep. So mu is for IgM, M is in mu. Delta, IgD. Gamma, IgG. Epsilon, IgE. Alpha, IgA. Those are the Greek letters that roughly correspond to M, D, G, E, and A.
Epsilon was for IgE.
E.
What was for IgA?
Alpha.
That makes sense.
Yep.
IgM was mu, IgD was delta, IgE for epsilon, IgA for alpha. I feel like we're missing one.
IgG for gamma. Yes.
So on our exam, are you gonna have the mu, gamma, alpha symbols, or is it gonna be like IgA?
I will use those as appropriate. So yes, you should know that mu and creates IgM using the mu heavy chain.
Okay.
I realize most of us have not studied Greek or Latin. Okay, I saw a set of eyebrows raised there, which means somebody has studied Latin.
Yep.
A couple people. They didn't even offer Latin in my high school, so I missed out on that. I wish I had been able to take it. It would have been very helpful, but I did not. So these are the Greek letters that are no longer in use for normal, like they don't take part in the English alphabet, but they are still seen in other alphabets around the world. Some other alphabets around the world, including modern day Greek. All right. Everybody with me so far? Good. This is the structure of a basic antibody. Because you'll notice where this actually says V-pre-B1 over here. This is a temporary or placeholder light chain. And you get this one before light chain recombination, but after heavy chain recombination. Structurally, it's fairly similar. It uses slightly different genetics, and all of them are the same. So it doesn't really matter. Like, this is just going to serve as a placeholder and enable survival signals to come through those Ig alpha and Ig beta. Survival and mitosis signals to come through Ig alpha and Ig beta, because again, the cytoplasmic tails of the B cell receptor were too short for them to signal on their own, so they needed those accessory molecules, Ig alpha and Ig beta. Right. Each B cell is going to make one heavy chain and one light chain, and only one. There's a reason for that. It's called allelic exclusion. And this means that only one of the two alleles, only one of the two chromosomes gets used. That is, gene copies from only one of the two chromosomes in a pair. Which chromosome gets expressed initially? It's random. So flip a coin, heads or tails, chromosome A or chromosome B. It doesn't matter. If the first attempt results in a functional B cell receptor, the cell shuts off further recombination. So only that first allele got used. If the first attempt is not functional, then it'll try again and the other chromosome and attempt to make the second copy work. And if it can't do that, it failed at both attempts, then it goes through death by neglect. So initially, your first B cell receptor, you're going to have the newly recombined heavy chain that was successful either from either one of those two chromosomes. And the initial constant regions that it's always going to choose, the first one that's expressed is always mu. So your first antibody types or B cell receptor types are always IgM. That makes things a little bit simpler. And remember, that thing is initially going to get expressed with one of those precursor light chains, the V pre-B. And eventually, if light chain recombination is successful, you'll get that replacement with the real heavy chain, and I'm going to call that one mature. Okay, I can barely fit that word in there, but that's what that's supposed to be, is mature in blue. Usually, if it gets through heavy chain recombination, it can do light chain recombination. So the probability that it's going to be able to make a heavy chain but not a functional light chain is very, very low.
The surrogate light chain, the same thing?
Surrogate light chain is the same as the precursor. Okay, so this is the entire total pathway. Starting from a progenitor B cell. This is a B cell that has not yet done any recombination whatsoever. It first starts off by doing DJ recombination. If it gets through DJ recombination and that part was successful, it will proceed to VDJ recombination on the heavy chain. If that is not productive, it can try again on chromosome number 2. If that is unproductive, cell death occurs. But let's assume that you actually got a productive recombination on either one of those attempts, either the first attempt or the second attempt. Okay, then you make a new heavy chain to go with your nice new, the rest of your variable region, the tips of your antibodies, the heavy tip of your antibody will combine with the mu region. You'll go through several rounds of mitosis because you get survival and mitosis signals through this pre-B cell receptor, which actually has the mu light chain on it, or the surrogate light chain on it. And then you can either try recombining the kappa or the lambda light chains in mice, which is what this particular diagram is set up for. Mice try the kappa light chain first. Humans don't. We can try either kappa or lambda. It doesn't matter as much for us. But in mice, they try the kappa light chain first. If that is non-productive, the cells eventually will die from neglect. If this is eventually successful or still unsuccessful, then it will switch over and try light chain lambda recombination on both of those 2 alleles. And if none of those 4 possibilities are correct, then eventually it dies by neglect again. So that's this whole basic diagram. It's got 6 different stages where it's got a chance to make a functional receptor. And if it fails at all of those 6, especially if it fails in these first 2, it dies. But if it fails after that, it's got 4 more options to try to get through and make a working light chain. This is why if it makes a heavy chain, it almost always successfully makes light chain recombination, is because you've got 4 shots at it.
Is that in humans too? Like will we do like either and only have 2 chances? Or
We still basically get 4 chances, yes. So that occurs also in humans. Okay, so this is where we left off last class. We were talking about receptor editing and what happens as these B cells mature, in particular the B cells, because that's where we decided to start this particular journey. So once you've rearranged a heavy chain successfully and you've rearranged a light chain successfully, what's supposed to happen next is you're supposed to get several rounds of mitosis, and this is because of signaling through that pre-B cell receptor. Those rounds of mitosis go on again after you get a complete mature B cell receptor. But you've also got to test these cells for self-tolerance. That is, the ability to not react against your own tissues. So if this actually happens, if your B cell does bind to self-tissues in the bone marrow or in the spleen, this recombination machinery can be turned back on to try and edit the original arrangement to salvage it and make it into a non-reactive B cell receptor, or to at least inactivate it so that it doesn't go on and continue to cause problems. So either way, either that cell dying or successfully recombining another immunoglobulin protein, either of those outcomes is preferable to autoimmunity. It's going to be one of those mornings. Okay. And apparently, it actually defaults back to attempting to do the recombination at the light chain if it has to try to edit this receptor and make a slightly different version of the same receptor or the same basic receptor. So the heavy chain remains consistent, but it'll try to recombine a new light chain. If that is unsuccessful, it'll move over to the second allele on the other chromosome copy and try to recombine there. If neither of those are successful, what happens? Cell death, apoptosis. Yep, that is absolutely correct. So this editing process really works basically the same way as the original recombination did. It's just a second round of it. So what they're showing in this particular diagram is you start off, you've got your V cluster genes, your J cluster genes, and the constant region, which down there, that one should be at this stage. Does anybody remember what the first one is? D, not quite. There is no C. Which constant region? Your choices include mu, delta, epsilon, alpha, gamma. Okay.
What's the question?
What constant region is used? The mu constant region. We're still using the mu constant region. The other constant regions don't get used yet. So this V cluster and J cluster, they show an initial recombination event between cluster 3 in the V region and cluster 3 in the J region. That makes this nice little joined 3-3, and this was the initial attempt. If this one is autoreactive, or if it's not successful, if it doesn't actually make a functional protein for whatever reason, then the cell can go back and try, even on this same chromosome, it can try recombining again. It can't do anything else with the 3, and 1 and 2 have now been lost, but it can take, because 1 and 2 were in between 3 and 3, so these two get lost from this process. But 2 and 4 are still available, and so the cell can actually go back and try this other combination, or it could try 1 and 4 to see if any of those potentially might actually create a functional immune response or a functional immune protein that is not self-reactive.
What happens to the 3-3?
The 3-3 gets clipped out and degraded in an excision loop, the same as it did during the initial recombination event. Okay, so your mature B cells express two different versions of antibodies, both IgM using the mu constant region and IgD using the delta constant region. And these are expressed pretty much at the same time. Mu comes first because that is the default one that is used most often, and that is for the first one expressed by all developing B cells. But once these cells become fully mature, they will also express mu and delta chains, so both IgM and IgD at the same time. And this is controlled by a splicing mechanism. So all of this is basically created as one long primary messenger RNA. So normally what you would have is a messenger RNA that codes for IgM and a second one that would code separately for IgD. That's not what we actually see in this system. What we actually see is just one long, big messenger RNA that actually codes for both it's got the variable diversity joining regions, and then it actually has both the mu region and the delta region all encoded in the same messenger RNA. So what it will do is it'll use alternative splicing, and it can either express VDJ mu, or it can express VDJ delta. So a little bit of that recombination-like effects can actually take place at the level of messenger RNA. And we talked a little bit about splicing and the process of splicing, but it's not totally dissimilar from what we saw happen in the genetic recombination. So there's basically some enzymes that come in, they'll clip out the intervening sequence of DNA, create a lariat loop structure, and that'll get degraded a lot like the excision loop from recombination at the DNA level did. So in this case, the RNA polymerase makes a very long initial transcript that contains the recombined variable VDJ stuff. That's all of this stuff. And then it's got the two possible constant regions, either mu or delta, and which of those constant regions it selects depends entirely on processes that I don't understand as well as I'd like to, but I want to call it kind of random. Mu seems to be favored. It seems to express more IgM than IgD, but it doesn't really matter. Either one can be created out of this process. IgD expression, though, seems to be most common in secondary lymphoid organs like the lymph nodes and the spleen. So there's some research that implies that it might actually be that it might serve as an adhesion molecule to help keep these B cells physically located in the lymph nodes or spleen once they're functionally mature and have a full functioning B cell receptor. Later, after these cells encounter antigen, you'll turn on the entire recombination machinery yet again and remove the mu and delta constant regions so the cell can actually express either IgG, IgA, or IgE. That's called class switching, and it's one that we'll talk about later on this semester. So this term that I have underlined over here, polycistronic, that means that your messenger RNA contains multiple genes. And in this case, it's got the genes for both mu and delta. Polycistronic messenger RNAs are not uncommon in our cells, but they're a lot more common in bacteria. And it's a little bit more complicated than just IgM and IgD regions. This messenger RNA splicing mechanism also controls whether the cell produces membrane-bound IgM or secreted antibody version of IgM. So that process is also regulated here at the splicing level. So basically what you've got is this big long messenger RNA. You've got the V, D, and J regions. You've got a C mu region, and you should have a region both with and without a cytoplasmic tail. So depending on how it chops up this particular C region, you've got with or without a tail, the cytoplasmic end. And then you've got the constant region delta that also has with or without tail options. So kind of like 4 different gene segments that can be encoded based on this one single messenger RNA that give you 4 different possible outcomes. So at this stage, once a B cell is mature, it's made a successful B cell receptor, it's put that at the surface of the cell, it's getting its pro-survival signals, and it's doing all 4 of these things at the same time. So it's going to make a little bit of the membrane IgM and a lesser degree of soluble antibody IgM that's secreted from the cell. While the cells are still mature but naive, they don't make a whole lot of secreted antibody. And then it'll also be making IgD, especially when it's in the secondary lymphoid organs like the lymph nodes or the spleen. It'll make both membrane-bound and soluble antibody versions of IgD as well. Yes?
So it uses both IgM and IgD, but doesn't use the others, I guess?
It does not use the others at this time. Those only turn on after antigen exposure.
Okay.
So, what about the T cell receptor? It works a lot like the B cell receptor. The structure of the T cell receptor is much simpler than that of the B cell receptor, so you've really got just variable region and a constant region. So you've got those 4 immunoglobulin loop domains as this thing is constructed as a primer, And what you're looking at are the kind of diagrammatic version of that in A, and then B, you've got the protein structure, if you were looking at it kind of from the top down. And then in C, as you zoom in a little bit more, those are those complementarity determining regions, or the parts that are specifically supposed to interact the most with antibody. So this is the last little part of what we need to learn about, is how T-cell receptor editing is done. The whole process basically works exactly like B-cell receptor editing. It's just slightly different because of the structural differences between these two receptors. B-cells were discovered, or the B-cell receptor editing process was discovered first, because it was a lot easier for them to track down soluble antibodies and look at their protein structure and then find cells that matched. T-cells don't make antibodies, and they don't secrete T-cell receptors. Those are only membrane bound. So it made these cells a lot harder to isolate, identify, and study compared to the B. And they had to make some interesting assumptions about what this receptor should look like and how its genetics should behave. First and foremost, the T-cell receptor had to be expressed uniquely and only on T-cells. So this had to be, they knew they were looking for a protein that was only on T-cells, that was not also found on B cells or NK cells or anything like that. It had to be specific for T-cells. They knew they were looking for a membrane-bound protein. So for it to be membrane-bound, that means that during the process of creation, this thing has to traffic through the endoplasmic reticulum and the Golgi. So they should be able to isolate the messenger RNA for this particular membrane-bound protein that is unique to T cells from ribosomes stuck on the endoplasmic reticulum, since they know it has to go through that pathway to end up on the cell membrane. That was a particularly brilliant little piece of thinking that somebody did there. Looking back at it, it's like, well, okay, that's logical and that's obvious, but it was not obvious at the time. Frankly, it's not obvious to me a lot of the time. Like, sometimes I look at things and I look at solutions that people come up with to questions, and I can see how they got there, but I know that I never would have thought of it. Let me put it that way. For T-cell receptors, you're faced with the same problem though, and that is the problem of diversity. So once they understood how B-cell receptors were being generated, they said that this is probably going to be a very similar mechanism for T-cells, because you still have to take a limited amount of DNA genetic information and still be able to make millions of different combinations of that, so that we can detect millions of different types of pathogens. They were right about that. They also postulated that the T-cell receptor genes, a lot like most of the other immunoglobulin genes and antibody-related things, should have variable regions and constant regions, which is exactly what we have seen. So the quick reminder of what the structure of the T-cell receptor looked like compared to the B-cell receptor: it was short and stubby. You've only got the variable regions at the tips and one constant region, and then the cytoplasmic anchoring tails, which are a little bit too short for cell signaling. Understanding the structure of this protein was really important to actually discovering the genes that are coded for it. So the protein structure came first, and then we found the DNA that actually made that. And that kind of makes sense. The protein is going to be there and it's going to be stable, but the DNA has to do all that recombining stuff, or at least they predicted that it did. So we knew that we would be looking for clusters of DNA in our genetics that didn't exactly map to the final protein. Like there would be other stuff in between these little clusters. And what they did was they actually used monoclonal antibodies that bound to specific parts of the T-cell receptor. So they actually generated antibodies to T-cell receptors and then used those to try to purify T-cell receptors. And they found out that it was a heterodimer, meaning it's got two subunits that are different from each other with an alpha and beta chain. There is another version of T-cells. It's not just the alpha and beta T cells, although those are the ones we're going to talk about the most this semester. There's also a set of cells that make gamma delta T cell receptors. We're going to talk about those a little bit, but a whole lot less. Most of our classical T cells are the alpha beta subtype. And they actually discovered the beta chain gene first. And sure enough, consistent with their predictions, what they found was that there were, in fact, multiple gene segments. These actually appeared to have the exact same type of structure as for the B cells. So VD and J recombination occurs for these as well, even though you don't necessarily have a light and a heavy chain sequence. So what you've got there is a very limited number of D diversity regions, but you've got a lot of V regions, variable regions, and a smaller number of D and J regions. While they were looking for the alpha chain, though, they accidentally discovered the gamma chain genes first. And this was actually interesting because it told us a little bit of something important about how these T cell receptors actually get generated. Normally, if the cell recombines, it's going to try one of two combinations, either alpha, beta, or gamma delta. And it's actually going to try basically all of these simultaneously, and it's a race to see which one gets done first. Typically, you get the beta gene that recombines first, and then alpha. Whereas gamma delta must occur simultaneously. The fact that gamma delta have to occur at the same time, it means it's a little bit less likely for those to be successful. And so you get fewer gamma delta T cells than alpha beta T cells. There's something weird about those gamma delta T cell receptors also. They recognize unusual antigens, such as lipids and glycolipids. This part is really important, so make sure you get that in your notes. Gamma delta T cell receptors do lipids, not proteins. They also do not need MHC because they're not looking for peptides presented on MHC. They're actually just straight up looking for lipids, so they don't actually need a presentation pedestal to help with that. Alpha beta T cell receptors, those actually recognize peptides in the context of or presented on MHC, which is the topic of our next chapter, which we will probably actually start today. So these T cell receptor genes undergo the same basic process using the RAG1 and RAG2 enzymes. But this means something very important. So previously, I mentioned that people and animals that are born with mutated versions of RAG1 and RAG2 cannot make functional B cells. They also cannot make functional T cells. This is catastrophic. That means that these people, these animals, have no adaptive immune system at all. They are completely reliant on their innate immune system, and it fails. People who are born with RAG1 and RAG2 deficiencies or mutations that are severe, they actually have something called severe combined immunodeficiency. Or SCID, S-C-I-D. And these people have to basically be kept in a bubble, literally a bubble. They cannot interact with the outside world. The only way they can survive is if they do not contact pathogens of basically any type. That is extremely difficult to do in a modern context. They have to be kept in a sterile environment. That means food has to be sterilized, clothes have to be sterilized, air has to be filtered, all of it, for these people to survive. They do not have long lifespans. Most of them do not make it to adulthood. Baby animals that are born with this, they just don't make it. They die very quickly. They get an infection almost immediately and they die. If you ever go work in immunology research and you work with animals in the lab that are skid mice, these usually are kept in special facilities. You may have to like shower in, dress in like full gear and handle these mice very carefully and very, very separately from the rest of your mice. So it's usually a shower in, shower out situation. It's unpleasant, but not unsurvivable. Like, it's not the most fun thing to have to shower like twice a day at work, but you can do it. It's not that big of a deal. I've done it when I worked with mice. However, the good news is you've got the same signal recognition sequences, RSSs, and same enzymes as used in the alpha, as in the beta or B cell recombination. Yes.
What are these, the RAG1 and 2 things again?
RAG1 and 2 is the enzyme that does the cutting of the DNA and makes that initial single strand nick.
Okay.
This is also what gets those 12 and 23 base pair spacer sequences to hold that loop of DNA in place. Okay. So what's different is that B cells basically go through this sequential process where they rearrange the heavy chain and then they rearrange the light chain. T cells, they are going to try the alpha, delta, and beta genes all at the same time. You'll notice that one's missing. Gamma, yep. Only if you get a successful delta chain will the cell attempt to recombine a matching gamma chain. But basically, you've got this same type of idea. You get rearrangement at either the delta or the beta gene first. If that is successful, the cell will try to recombine alpha or gamma.
What does that say?
Second.
Oh.
That was supposed to be a 2. ND. If the cell is actually successful at making a beta chain, since most of our T cells are alpha beta chain or alpha beta T cells, let's talk about those first. If the cell is successful at making a beta chain, it will go ahead and stick that on the surface, just like it did with like the B cells did with the heavy chain. It will use a surrogate alpha chain, just like B cells used a surrogate light chain. And as long as the cell gets pro-survival signals through that temporary placeholder T cell receptor, the cell gets happy and it goes through several rounds of mitosis, just like the B cells did. It's the same basic process, except most of this process takes place in the thymus for T cells. After that, after the mitosis rounds, then it will attempt to recombine an alpha chain. If that is successful, the cell goes through, or T cells go through 2 rounds of selection, positive selection and negative selection. It has to be able to bind to peptide, and it has to not bind too strongly, especially to our own self-peptides. Yes?
B cells do the same.
B cells do the equivalent, yes. We'll look at those processes in more detail too in the next few chapters.
T cells use you said IgA, IgD, and IgG with alpha, delta, gamma, and B cells use IgM, and IgD.
T cells do not use Ig anything.
I thought you said alpha, gamma.
Alpha, beta, or gamma delta, but those are not those are not antibody proteins. Different, structurally similar. They're still related. They have the immunoglobulin domains, but they're not part of the B cell antibody gene cluster. So you've got Ig alpha. I know there's a lot of alphas and betas and gammas, but in this case, these are totally separate genes that just happen to have the same name.
Naming it in biology is confusing.
Yeah, naming is a problem. Unfortunately, there's only like 26 letters in the alphabet, and we ran out of. There are too many things.
So,
All right.
You said T cells don't use Ig.
T cells do not use the same genes as B cells. They have a separate gene cluster. This is a totally separate gene cluster.
I thought the Ig stuff is what they bind to. Binds to stuff.
Immunoglobulin specifically refers to the structure, usually of the B cell receptor or antibody that is soluble in the bloodstream. It also is a term that is used to refer to the specific loop's domain structure that is stabilized by disulfide bonds.